Mhm.
Um, I'm very excited to share with you um, I would say, the main focus on my lab for the last 20 or so years, and also share a few new stories. Um, but I'd like to start actually by talking about the main clinical and biological motivation behind our work. Okay. So I think many of you probably heard about the fact that drug resistance is one of the biggest obstacle to effective cancer treatment. There is a situation where there is a diagnosis, there is treatment that works very well. The treatment leads to remission, to shrinkage of the tumor, and then after days, weeks, months, sometimes years, there is a relapse. And some cells within the tumor survive the treatment and can develop into new tumors. And a lot of focus is going into understanding the acquisition of new mutations or genetic differences between cells that allow them to escape the treatment. But we're looking at it from a little bit of a different angle, which is trying to understand if there are differences that are not necessarily genetic, but even if you take genetically identical cells and you treat them with some drug or with radiation, you still get some cells that can be resistant, and these could be the cells that will later develop into tumors. Because we know that even in the lab, when we take human cells or mouse cells, cancer or normal, and we treat them with a drug that is supposed to kill them all.
Mhm.
Many times we see a mix of behaviors, right? We would see that some cells will indeed go through cell death, which is apoptosis. Some will senesce, which they enter these permanent cell cycle arrest, and some will recover. Right. So, as much as possible, we are doing our research at the resolution of single cells in order
Mhm.
to understand these decision making at the single cell level. Okay, so that's one of the key things in our approach. The second is the idea to look at things temporally over time. And I think it's very easy to explain the motivation behind that. So imagine that you have a situation that you want to learn how a drug works.
Mhm.
And you test the impact of the drug on cells, and you say it's before the drug, right? The cell was at this state, right? And then after the drug, it switched into a new state. And you measure some factor, it can be anything, your protein of interest or localization or anything. You say before the drug, the level of the factor was very low, and at time point A it was very low, and at time point B it was high. And then you look at three other drugs and you see the exact same behavior that at the original time point it was low and then it was high. You may conclude that these three drugs have similar mechanism of actions because they did the exact same thing to the factor that you're measuring.
Mhm.
However, if you look at the thing temporally and not just at the snapshot before and after, you
Mhm.
may realize that this factor reached from A to B in very
Mhm.
different ways, right?
Mhm.
Maybe in the response to drug
Yeah.
one, it just immediately increased and then
Mhm.
plateau.
Mhm.
Drug two, it was a switch. Drug three, it oscillated, right? So looking at things at high temporal resolution can reveal how things change over time. And why this is important? Well, first of all, if you understand the dynamics of the system, it can teach you a lot about the network structure, all the feedbacks and connection that lead to these dynamics, right? How these dynamics are encoded. And it can also teach you a lot about the cellular outcome, how these dynamics are decoded in
Mhm.
order to trigger different outcomes. Okay. And so we are really obsessed about dynamics and about the idea that cellular information is transferred through modulation of protein dynamics. And once you think about protein dynamics, you realize that it's so much more rich than this thinking about levels of a protein, right? If
Mhm.
there's a little or a lot, you look at many different parameters such as frequency, amplitude, integrated signal, delay, duration, and so on. And so in our lab, we're really obsessed and interested in understanding how cells encode information using these parameters and how cells decode this information to trigger the outputs. And so the big umbrella is that we believe to understand fundamental processes in biology, as well as why a subpopulation of cells escape drug actions. We need to investigate the dynamics of cellular responses at the single cell level. All right, that's the big umbrella of the lab, looking at dynamics and looking at single cells. And with this approach in the last many years, we looked at many different cellular pathways, regulation of cell cycle, DNA damage, cell growth, cell death, differentiation, oncogenic induction. But the pathway that has been the main focus of the lab, and this is the one that I'm going to talk about today, is the p53 signaling pathway. So let me tell you what you need to know about p53 in order to understand our work, because there's a lot has been published about p53. But first of all, so it's a tumor suppressor, and it's a transcription factor. So it responds to many kinds of stress. Okay, when cells feel stress, and it can range from DNA damage and hypoxia, other kind of stress, p53 levels increase, and then p53 activate the transcription of many target genes, and these target genes participate in executing a cellular response and affecting the cellular outcome. So for example, in response to DNA damage, p53 level go up, and then it assesses the damage. It leads to a transient arrest and recovery, or it can activate cell death, or it can activate senescence, okay, which is cell cycle arrest. And a big question in the p53 field has been how this one single protein can choose between these alternative outcomes, which, if you think about it, they're actually conflicting. Like a transient arrest, this means recovery, but cell death and senescence are what we call terminal fates. The cells cannot recover from them. So it's the same factor can allow the cell to grow, or it can stop the growth of cells. And why this is important? Well, first of all, in cancer, about 50% of human cancer have mutations in p53. And so when p53 is mutated, damaged cells don't arrest, don't die. They can continue to grow and can develop into tumors. But in 50% of human cancer, there is wild type p53, right? And there is a very big effort, both in academia and in pharmaceutical companies, to understand how this wild type p53 works. And can we use this wild type p53 in order to push those cancer cells to cell death and to senescence? Okay. And so if we can understand how we can affect these decisions, we can first of all push cancer cells toward stopping growth, but we can also protect normal cells from dying in response to treatment. And so, as I said, we're not, of course, the only ones asking how p53 chooses these outcomes. And there are a lot of models out there talking about a threshold, a level, that if you have a little bit of p53, it allows recovery. If you have a lot, it will lead to death or senescence. Or models talking about modification, close translational modifications, that depending where it's modified, it will activate different outcomes or cofactors that bind to it. And so all of these things are true and they're correct and they're important, but it's very clear that there's still gaps in our understanding of how p53 chooses between these outcomes. And the main theme or question we had in the lab is whether cells use modulation of p53 dynamics to achieve specificity in this network, right? Does the dynamics of the p53 protein play any role in the decision-making of what will happen downstream in response to damage? Okay, so before we're going to look into how we measure p53 dynamics, one very critical thing about what controls its dynamics. Oh, I'm just going to summarize what I will tell you today. So I'll tell you about the approaches for studying p53 dynamics in single cell. What we discovered is p53 dynamics in response to DNA damage. Then show you two examples for how, when we study p53 dynamics, it can help us guide the design of combinatorial therapy. And a recently story about the dynamics of p53 post-translational modifications. Okay. All right. So now, when we talk about p53 dynamics, it's very critical to know that we are talking about the protein level. Okay. And p53 as I said, transcription factor, and it activates the transcription of a gene called MDM2, which is a negative regulator of p53. It binds to p53, degrades it, and sends it to degradation. Okay, so they create this very classical negative feedback loop with two arms with different time scale. There is this transcription arm that is a slow rate response, and there is the protein-protein arm, which is a fast rate response. Okay, and these two feedbacks, basically the parameters of these two, will determine the dynamics of p53. And even with just very simple feedback, we can get oscillations, we can get one pulse, we can get a continuous increase. Now, it's important to note that the transcription, the level of p53 mRNA, the expression of the protein, is constant. It doesn't change after DNA damage. The things that is changed is the stability, right? So in happy cells with no DNA damage, p53 is expressed all the time. It's expressed and it leads to MDM2, and MDM2 degrades it. And this is what keeps p53 levels low. Okay, it's being expressed, expressed, degrade. Once there is DNA damage, then the degradation, this arrow is being inhibited, and this is what leads to induction of p53, and the stability, the half-life of the protein increases. Okay. And so I told you we like to measure it in single cells. So the technique that we use is to, we work with mouse or human cells, and we put a fluorescently tagged p53 and create clonal cells that all of them are genetically identical. And then we treat cells, we either irradiate them or treat them with a drug. And then we run movies. We basically follow the behavior of cells for hours and days. This is an example for the images that we have where you see this is a cell. This is the boundary of the cell, right? And this is the nucleus. Wow. And the green color is basically how much p53 this cell has, right? And this is an example of lung cancer cells that were treated with a chemotherapeutic drug. And I'm going to play this movie, and you'll basically see that the level of p53 goes up, increases, and then until cells start to like pop. This is basically cells going through apoptosis because of a lot of DNA damage. Okay, so we follow p53 levels in individual cells through the fluorescence intensity of the fluorescent protein that binds to p53. So one of the things that we discovered several years ago is if we take, it's either cancer cells or normal cells, any cells with wild type p53, and we treat them with gamma radiation, same levels of radiation that cancer patients receive in the clinic, then p53 go through these series of beautiful oscillations, right, that are found really fascinating. For many hours and sometimes even for many days, right? What was also fascinating about these oscillations is that we could find cells with different number of oscillations. Some cells have less number of these pulses and some have more. And then by looking at both the breaks in the DNA and p53 oscillations, we discovered that the number of p53 pulses increases with the amount of DNA damage, right? So think about it like this, a digital response, and it's like a while loop. So while there is DNA damage, pulse, then pause, repair the damage, and then check, is there still DNA damage? If yes, go through another pulse, and if not, just stop pulsing, right? And what we love about this system is that we don't get any damped oscillations, right? You don't pulse and then it's just damped. It just stop. If it stops, and then it just go back to basal level, right? And we've worked for many years to understand the molecular mechanism leading into that, and develop some models to explain how this dynamics is being encoded. We also found that the periods of p53 oscillations is extremely robust, right? It's about 5.5 hours, right? And it's fixed across different doses. It is fixed across cells. It's fixed across different cell types. It's very robust, but the amplitude is quite noisy. And when we looked at these, noisy amplitude, we see that sometimes if there is a cell that has probably just by chance, right? Just by noise, like a series of low amplitude P53, then instead of these cells arresting, right, and staying arrested for a long time, these cells can actually escape. They can escape cell cycle arrest and proliferate. So meaning the amplitude of P53 is really important in order to keep cells arrested and prevent them from escaping cell cycle arrest. Okay, and we
Mm-hmm.
showed in that paper that it basically allowed this like a switch within in one of the target genes of P53 that allow this
Mm-hmm.
escape. And when cells escape, when they do these escape,
Mm-hmm.
then there is a switch in P53 dynamics, right? So
Oh.
if cells don't escape, they continue to oscillate for many hours. But if a cell escape because of low amplitude P53 oscillations,
Mm-hmm.
it trigger these pathway through the caspase 2 pitosome,
Mm-hmm.
which inhibits MDM2, and then P53 oscillations are switched into sustain. So
Mm.
you see oscillations, and then after that, you see a high sustained level of P53, which
Mm-hmm.
we believe it works as like a safe, fail mechanism. Okay, if a cell escape arrest, it wasn't supposed to escape, but it escaped. Now P53 is going to stay really high to make sure it will not escape again. And in the past, we found like artificial ways to change P53 dynamics in order to learn how it impacts the programs downstream. So it's relatively easy to do because there is a small molecule called Nutlin-3, and now there is a new generation of these molecules they inhibit MDM2.
Mm-hmm.
So you can irradiate cells, which lead to oscillations, and then you add Nutlin-3 to inhibit MDM2, and only by this you can switch P53 oscillatory behavior into what we call sustain. Okay, and then this provides a tool to see how these dynamics translate into gene expression into cellular outcomes. And what we found in the past is that if we only change P53 dynamics using
Mm-hmm.
an MDM2 inhibitor from pulsatile into sustain, we change the fate of the cells, we change the outcome. Pulses of P53 allow recovery and growth, but if we switch them into sustain, we push cells into senescence. Okay.
So Galit, then that escaped cell due to the low amplitude of the P53 will be sustained, then the decision will be the cellular senescence.
Yes, so the once they escape, they will switch into sustains and then they will senesce. Yes.
Okay, gotcha. Thank
Yes.
you.
Yes.
So I have also a related question. So
Yeah.
cell may be able to use sustained, but why cell do not use the pulse further to uh, to resolve the problem? The cell cycle arrest has been escaped.
You asking why naturally cells don't have sustained P53?
No, I mean cell may have two options using more pulsed P53 or sustained, but why
Yeah.
cell pick the cell sustained dynamics when the cell cycle arrest is canceled or escaped?
Oh, so we understand, why is a very difficult question, right?
Yeah, I
Why go
see.
into evolutionary? What's the advantage of that?
Hmm.
So I can tell you how it's happening, right? We
Hmm.
understand the molecular mechanism that in which inhibits MDM2. The evolutionary benefit of that, I think that's quite difficult to
I
answer.
see. Thank
Why
you.
they're not just continue to
Hmm.
oscillate instead of that they switch to sustain.
I see. Thank you.
Yeah.
Can I ask one question?
Yes, of course.
I would like to uh the the connection between the lower amplitude of P53 and the escape time is I missed that part. Could you go back to that slide to
Yep.
yeah, I could you explain again the why lower amplitude is
Uh,
important in this graph?
I think it's not about the why. When they reach the lower amplitude, they just escape.
Okay. Then again, my further question then, what is the meaning of the escape in cell sense? Is the escape from the cycle? What is the meaning that? It's just the escape from the cycle means just the
Senescence.
apoptosis, senescence like that?
No, I escape from arrest. So these
Mm.
cells,
Mm-hmm.
they because of the DNA damage they
Okay.
stop cycling, they stop proliferating.
Yeah,
Okay.
yeah, yeah,
So
yeah.
that is called cell cycle arrest.
Yeah.
And they escape from cell cycle arrest, meaning they go back into dividing. You don't want them to divide because they have a lot
Uh,
of damage, right? So they escape the arrest by dividing.
oh, thank you, thank you.
Okay. Any other questions?
Yeah. Yeah, you can go ahead. Thank you.
Okay. Um, So, and another actually really interesting thing, if you remember, I told you that the oscillations are really,
Mm.
the period is robust.
Mm-hmm.
But it's very interesting that
Oh.
actually in mouse,
Mm.
the period of oscillation is shorter.
Shorter.
In
Oh.
mouse cells oscillate quicker, and
Oh.
it's like exactly the half of the period in human. In human, it's about 5.5 hours, and
Uh-huh.
in mouse, it's about 2.7,
Oh.
which I find extremely fascinating.
Yeah.
We don't understand why. Like,
Okay.
what is the advantage of the faster oscillations in a mouse compared to the human? If anyone has idea like why this is happening, I would
Uh-huh.
love to hear that. Like, again, we understand the mechanism, we study the feedback in the mouse, we know that they are faster and stronger, but what's the benefit from evolutionary perspective? We don't fully understand. But we did have one study in which we looked at P53 in vivo. We don't have a live reporter to look at P53 in mice, so we took snapshots, right? We basically did use antibodies to look at P53 levels in different tissues after radiation. This is just an example of the small intestine with an antibody that's showing P53. The green color is P53. I'm showing it just because I think it's really, really pretty. But if we follow P53 after radiation in different tissues, we see that before radiation, there's almost no P53 in those tissues. After radiation, tissues induce P53. But the interesting thing is that the dynamics of P53 is different between the different tissues. So let me show you here's an example. If we look some of the tissues, we see P53 levels go up and stay high in the thymus, spleen, bone marrow. Marrow needs to be here. But in other tissues, small intestine, large intestine, skin, we see something that looks like oscillations or maybe just like a wide pulse, okay? And what we realize in that
Mm-hmm.
the tissues that
Wow.
consider to be more sensitive,
Mm-hmm.
the dynamics is more sustained.
Mm-hmm.
And the tissues that are more resistant, the dynamics is more temporary or oscillatory. We can also look at some target genes of P53. Here's MDM2, the negative regulator. P21 is an important protein leading to cell cycle arrest. PUMA is an important protein leading to apoptosis. And you see the same behavior, spleen and thymus, which are very sensitive. The dynamics is going up and then plateau. But in small and large intestine, which are less sensitive, we see oscillations. Okay, so there is a correlation basically between the tissue sensitivity and the dynamics of P53. And then in this study, we took it one step further and we looked at the treatment of a tumor after we changed P53 dynamics. So we took a mouse with a tumor that we put in the mouse, and we compared how this tumor behaved if we irradiated, that leading to oscillations of P53, or if we irradiated and added an MDM2 inhibitor that
Mm-hmm.
leads to sustained P53. And we see that just by changing the dynamics of P53, we lead to significant shrinkage of the tumor. Okay, so even in vivo, dynamics of P53 matters. And we like to think about the dynamics as a target, right? Instead of thinking about targeting a protein or targeting a factor, we're targeting the dynamics and we're very accurately changing it in order to control the fate of the cells. I showed you so far the response to gamma radiation, but throughout the years, we tried different treatments and we found that P53 dynamics depend on the input. So in response to gamma, it oscillates, but in response to UV, that leads to a different type of DNA damage. You just get one big pulse. And I want to show you a few slides about a study about cisplatin. Cisplatin is a chemotherapeutic drug that is one of the standard drugs in the clinic, and that leads to a different type of behavior. P53 levels just go up, 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 up. This study actually started by looking at cells. These are colon cancer cells treated with IC50 of cisplatin. If you all know, IC50 is a concentration of a drug in which 50% of the cells die and 50% of the cells survive, okay? And this is one of the most common measured by, pharma, biotech, is like the IC50. And we are really fascinated in understanding why. What is it that 50% die and 50% survive, right? Is this something that is completely stochastic? Or is there a reason, right? There's all genetically identical cells, still one die, one survive. Is there a reason for that? And if we understand the reason, can we trick the cells so that we use a drug in an IC50, but we lead to 100% cell death, right? Because you don't want to use very high concentration of a drug that can be cytotoxic, okay? So we took these colon cancer cells with a fluorescently tagged P53, treated them with cisplatin at the IC50, and this is the movie I'll show you. And you start to see these greenish color, right? This is P53 going up. And when you see these white things, this is actually cells that died. So we start to see cell death. Some cells are dying. P53 continues to go up. And this is about, a 72-hour movie. And that's the last frame of the movie. And you look at this frame, and it seems like a mess. Basically, you have everything here. You have a lot of dead cells, right? These white blurbs. You have cells that seem like they survive, but they have low levels of P53. But then you have a lot of cells that survive and have quite a lot of P53, right? And you start to wonder, okay, why this cell survived and this cell died? And we know absolutely that death in these cells really depends on P53, right? If there's no P53, there's no cell death. And so we started to draw basically the curves of P53 accumulation. We looked at hundreds of these. I'm just showing you a small number so it will be more clear. And then we can color them based on whether a cell die or a cell survive, okay? You immediately can see that there is a difference in the rate at which P53 is induced. And the field really likes to talk about something called the apoptotic threshold. So to say, okay, once P53 crosses this level, then the cell will die. And so we try to find the apoptotic threshold, and we realize that there's just no way, no place we can put a threshold that will explain the data, right? If we put it here, let's say we say, okay, all the cells that die need, in order to die, you need to cross the threshold. This explains the dying cell, but it doesn't explain the surviving cells, right? They cross it and they still survive. If we push the threshold up, right, and we say it needs to be beyond here, it explains the surviving cells, it doesn't explain the dying cells. So we couldn't find a simple static threshold that would explain that, okay? And then we realized that we are working with what we call a dynamic threshold. With time, cells need more and more and more P53 in order to die, all right? At the beginning, if you look at the first few hours, just a little P53 is enough to die. But then if you look later, you need more and more and more P53, okay? And so this is explained why if a cell accumulates fast, it can die with less P53 than if it's accumulate later. And through a lot of different experiments and literature and trial, we understood why the threshold moved with time. And the reason is because, we are measuring P53. But in addition to P53, many other things are happening in response to cisplatin. And there's a second arm that is induced in response to the drug, which is a group of proteins called inhibitors of apoptosis, okay? And we're not measuring these in live cells, but they are happening in live cells. And so when we add cisplatin, we have the P53 arm that pushes cells towards cell death and apoptosis, but then we have the anti-apoptotic arms that competes with P53, okay? If p53 is induced very quickly, it wins. But if not, with time, you have more and more and more inhibitors of apoptosis, and you need more p53 in order to fight that, right? And leads to apoptosis. And so if we use, for example, cisplatin that leads to 50% of cell death, there are two ways that we can manipulate the system and trigger more death. One of these ways is to add an MDM2 inhibitor, like the one we used before.
Mm-hmm.
What this does, it accelerates p53, right? It
Mm-hmm.
doesn't change the threshold, but
Mm-hmm.
it makes all the cells induce p53 faster, and you can get with IC50 close to 100% cell death. Okay, so one way you can just accelerate it the rate. The other way, which I find even more exciting, is if you use a drug that inhibit the other arm, right? There is a drug, we worked on this together with Novartis, and they, around that time, actually developed this drug. This drug inhibits the anti-apoptotic arm. So if you get rid of the anti-apoptotic arm, suddenly you became the threshold static, right? It's flat. And now all the cells just need to cross one number in order to die. And again, with an IC50 concentration, we can get close to 100% cell death. And any questions related to that before we move to something else?
Yeah. Any question? Yeah. Oh, yeah. To me, it's very clear. Wow.
Okay.
Yeah.
All right. So I want to show you one more examples of how the dynamics help us guide combinatorial therapy, and then I'll tell you a little bit about a new story. So this is related to this kind of dynamics, which also is really fascinating. In response to MDMX knockdown, let me tell you what is MDMX. So remember this from the beginning. MDMX is an oncogene that inhibits p53. So there is a high percentage of cancers that have wild type p53, but they have overexpression of MDMX. They have a lot of MDMX, and this MDMX suppresses p53 and doesn't let it do its job. So even though they're wild type, right, it can't function, right? So it can't arrest the cell cycle, it can lead to apoptosis, and this is why these cells develop into tumors. And so there are efforts to try and develop MDM2 inhibitors for those cancers that overexpress MDM2 so that you will let p53 kill those tumor cells. And what we found a few years ago is that if you take cells with a fluorescently tagged p53 and you you suppress MDMX, you get this super cool biphasic dynamics in which first p53 makes a big pulse and then you get a series of oscillations. I have to say, for someone even studying live cell imaging, one of the things that we always deal with is a huge heterogeneity between cells. Like sometimes cells look very, very different. This behavior is like so robust, like all the cells just do the exact same thing. We have one big pulse and then a series of oscillations. Super cool. And so we, in that study, we said, let's try to combine inhibition of MDMX with DNA damage, but add the DNA damage at different times of the response. Like let's add it during the big pulse and see what happened. And then let's add it during the oscillations and see what happened. Okay. And then in this study, we discovered that the interactions between these two treatments depend on the phase at which DNA damage was added. And so if we add DNA damage in what we call phase one during the one big pulse of p53, then we enhance cell death. Then MDMX inhibition and DNA damage, they're synergized with each other. They lead to better outcome of cell death. But if we add DNA damage at phase two, we actually protect the cells from DNA damage. We allow recovery and growth. So it was a situation in which the time interval between two treatments not only affected the synergy between two drugs, but completely flipped their interactions. Okay, it flipped it from being synergistic to being antagonistic, right? And we're really trying to push the notion, especially for those who are developing clinical treatments, to think about in combination therapy, to think about the sequence, what's added first, what's added second, and the time interval between them, because these things matter a lot. We already see it in a dish. I'm sure it's the same in patient. Okay, so we talked about these
Mm-hmm.
three things, and lastly, I want to share with you some new a new study about studying the dynamics of post-translational modifications. And first of all, the protein, the p53 protein, can be modified a lot, right? There are a lot of enzymes
Mm-hmm.
that can put all kind of
Mm-hmm.
modifications that you can think,
Mm-hmm.
phosphorylation, acetylation, methylation, everything,
Mm-hmm.
okay? And we have been very interested in understanding how the modifications and the dynamics of p53 work together to ensure a functional p53. And the question that we had is very simple, which is, he said, if we take p53 oscillations after radiation, as I showed you, and we look at the first pulse, the second pulse, and so on, are we going to find p53 with the same modifications or different modifications, right? Because remember, so far, we fused p53 to a fluorescent protein. So we just measured the total p53, right? The total protein, but we don't know where it's modified. And these modifications matter a lot for its
Mm-hmm.
function, right? So we
Mm-hmm.
said we want to see where it's modified in the first pulse, second pulse, and so on. The best way to do that is a technique called mass spectrometry, which you run the protein through a mass spectrometry and you identify all the peptide and you identify all the different modification sites. For reasons that I don't think we fully understand,
Mm-hmm.
p53 is one of the most complicated protein to do mass spec on.
Mm-hmm.
We've tried for at least, I think, 3 or 4 leading groups
Uh-huh.
in the US. I don't know, maybe
Uh-huh.
we need to try with Singapore,
Uh-huh.
um, but or with Korea, I don't know. But we haven't been able to do it to identify these. What we able to do is something that is called top-down mass spec. Okay, and I'll explain what top-down mass spec does. In top-down, you run the entire protein and you identify the weight of the whole modified protein. And what you get is the relative abundance of something that we called a mod form. So if we have a p53 modified in, let's say, these 4 residues, okay, we see all the distribution of the molecule that are modified here, and then we see how many molecules are modified at a different place or in a different place. We
Mm-hmm.
don't know what are the modifications. We can't tell what the modifications are, but we know how many species, how many different species, which are these mod forms we have, okay? And so we collaborated with Jeremy Gunawardena
Mm-hmm.
at Harvard Medical School
Mm-hmm.
and Neil Keller at Northwestern, who developed this top-down mass spec. We said, let's measure the mod form of p53 during the first pulse and the second pulse.
Mm-hmm.
If they're exactly the same, then we're done. We don't need to
Mm-hmm.
dive and understand. But if they're
Mm-hmm.
different, it will motivate us to know more. Okay.
Mm-hmm.
And these are the results. And just to orient you, so we took p53 from the first pulse, right? It's about two and
Mm-hmm.
a half hours, and the second pulse, which is
Uh-huh.
about 7.5 hours, and ran them through this mass spec.
Okay.
And this is the mod form distribution.
Uh-huh.
The peaks here is the least modified p53, and
Uh-huh.
here is the highest modified p53. And
Uh-huh.
this is the distributions of the mod form.
Uh-huh.
And one of the first things when I saw this, I was really surprised because
Sure.
remember a couple of slides back,
Mm-hmm.
the number of potential modification is
Mm-hmm.
huge. And then if you
Mm-hmm.
think about all the potential combination between
Mm-hmm.
them, it's an enormous number. But if you look at the number of mod forms here, it's relatively small, right? So there is something that prevent from all these modification to occur, and it's actually cluster into a relatively small number of mod form.
Mm-hmm.
Now, if you compare the first pulse and the second pulse, many mod forms look the same.
Mm-hmm.
But here I'm marking a couple of places in which we see there is a mod form in the first pulse, but not the second pulse, this
Mm-hmm.
one and this one as well. So this motivated us to look and try to understand which modification might be different
Mm-hmm.
between the first and second pulse.
Mm-hmm.
We use immunoblots, Western, for this. These are antibodies that recognize specific modifications on p53. And we looked at it at this, the first pulse and second pulse, okay?
Mm-hmm.
And so each one of these is a modification, a specific modifications. And you see like many of them don't change or they don't exist or they don't change, but there are two modifications at these two residue of the protein that are high in the first pulse and low in the second pulse, okay? It's at 373 and 382. And we can also look at their dynamics. So here, this is the Western of p53. This is the p53 protein level, go up and down and up and down. These are the oscillations. And then there's these two modifications that exist on the first pulse of p53, but not the second pulse of p53, okay? Part of the study was also to understand the regulation, what removes them, and so on. But I want to show you just like the final set of experiment that we did to understand whether it matters at all if p53 is modified or not. And so for this, we used mutants. We created p53 mutants that cannot be modified on these two residues. We also added another residue that we knew that cells didn't care about. And we also created a mutant that cannot be modified in both 373 and 382. So it's exactly the same p53. It just cannot be acetylated. It can't have these acetylation. And we created two cells, one with a wild type p53 and one with the 2KR. The 2KR cannot be modified, okay? And we said, let's look at we did RNA-seq. Let's look at all the target genes of p53 and see if they care, if it's modified or not modified, okay? And honestly, I expected to find that most target genes would prefer a wild type p53 and will perform less well if the p53 is mutated and cannot be modified. But the results, as always in biology, been a little more complicated. And this summarizes the results. What you have on the x-axis is the area under the curve, the mutant p53 that cannot be acetylated. And this is the wild type p53 that can be acetylated.
Wow.
And every dot is a gene. And so first of all, the gray genes that are very close to this linear one-to-one curve means that these genes don't care. They don't care if p53 is wild type or mutant, if it's acetylated or not acetylated. They're induced at quite similar level. The red ones here, these are genes that perform better if it's a wild type p53 compared to a p53 that cannot be acetylated, okay? These are the ones that we were expecting to find. But we also found this group of genes that are actually genes that prefer not acetylated p53. And they perform better when it's a mutant p53 that cannot be acetylated compared to wild type p53 that can be acetylated, okay? And so the way we like to think about it is that there is this interplay between oscillatory dynamics and p53 modification in order to temporally organize different processes in the cells. So if we look at total p53, right, we get two pulses that look exactly the same. But if we look at the acetylation, during the first pulse, it's acetylated. During the second pulse, it's not acetylated. Some genes don't care, but some genes prefer acetylated, meaning they're induced more during the first pulse. Some genes prefer not acetylated, they're induced more during the second pulse, right? So it's a way to temporally organize the response downstream. And, it's a little bit, I feel like, again, this is more like our speculation, but this may explain why we have this oscillatory behavior, because if you think about the advantage of oscillations is that you get rid of all the protein that was there during the first pulse, and you express new protein that can be modified or non-modified, and you don't just confuse the system, right, with the previous one that was there. So perhaps this is the way to solve this problem, but just expressing a new protein that can get new modification and at different time of the response do different things. So this study just came out at the end of last year. I thought I'll mention maybe two main things that we are thinking about now and doing now. One of them is a general question to understand how intrinsic and extrinsic factors shape p53 dynamics. We look at cell cycle phase, confluency, what happened if we grow cells at a higher confluency versus lower confluency, 3D structures. We work with spheroids. We also look at organoids, intestinal organoids, and look at how cell type impact p53 dynamics, trying to connect our studies a little bit more with in vivo and with the clinic. And we're really interested in understanding how p53 dynamics are translated into downstream processes. We have a tool now to modulate the frequency of p53. So we want to see if there is like a frequency modulation signature that affect the target genes. We look at transcriptional signatures and respond to different dynamics and how they are connected with cell fate. Also, the impact on DNA, the DNA repair itself, the actual repair of DNA. And, I describe a work of many people in many, many years, but these are the current lab members in the lab. We have also many collaborators within Harvard, outside of Harvard. Need to mention all the funding agencies and well, thank you for listening. I love to talk about our work. I prepared when I see my audience and can have a conversation, but it was important for me to leave enough time so that we can stop sharing, see everyone, and
Sure.
maybe have some discussion and questions. So thank you.
Thank you, Galit. Thanks so much. Thanks for the wonderful talk.